Hello, grade 9. Today, inshallah, we will start with chapter 7, Life is Cellular. Let's start with the first part of our lesson. We need to study the discovery of the cell. So, we need to start with the cell theory. All of you need to memorize all of the three parts of the cell theory, which is all living things are made up of cells. Cells are the basic units of structure and function in living things, and the last one, new cells are produced from existing cells. Okay? We need now to go back to the history of the cell and the early microscope, and we need to study the scientists that discovered the cell. Robert Hooke, which is the first person to use the microscope to look at a non-living specimen, he looked at a thin slice of cork, which is a plant material, and he found that the plant cell, or he called the empty chambers, he looked at cells. So this is empty chambers that he found it under the microscopes of the slice of cork, and he called these empty chambers as cells. So the first scientist who called the tiny things that is found in the plant as a cell is Hooke. Okay, he looked at the cork under the small microscope that he already created, which is called Hooke microscope. The second scientist, which is called Lewin Hooke, examines pond water and other things, including a sample taken from a human mouth, and he found the small tiny organisms, which he called them bacteria. Then we will move to Schwann, Theodor Schwann, which he stated that all animal cells or all animals were made of cells, and finally Mathias concluded that all plants are made of cells. This is how the cell theory developed and how the scientists defined the cells. Then Virchow, which he concluded that new cells could be produced only from division of existing cells. So without an existing cell, there is no way that the new cells will form. Okay, this is from Virchow. Let's now study the microscope. Most microscopes use lenses to magnify the image of an object by focusing light or electron. So we need to study the structure of the microscope. The microscope is made up of many units. First of all, an eyepiece, arm, fine focus knob, coarse focus knob. Then we have multiple lenses here, we call them objective lenses, and these objective lenses have many magnifications. Okay, we have the stage here, diaphragm, light and base, and finally aperture. We will put the specimen here, and we will turn on the light, and the light will pass through the specimen. Then the objective lenses will take the light, the transparent light, to the eyepiece, and the light will be magnified. The picture will be magnified. We need to know that we have multiple objective lenses, and these objective lenses have different number, and the eyepiece have a specific number. In the beginning, they found the light microscopes, and to see the cells under the light microscope, they need stains. So a typical light microscope allows light to pass through a specimen and uses two lenses to form an image. As we said before, the objective lens with the eyepiece lens. Okay? The second set of lens magnifies this image still further. Light microscopes are able to produce image of objectives only to a magnification about 10,000 times, because light waves are diffracted or scattered as they pass through the specimen. Okay, so we have here the objective lens. The objective lens magnification starts from 4 to 100, and we have the ocular lens, which is having a definite number, which is 10 magnification. So we will multiply the objective lens magnification with the ocular lens magnification to find the total magnification. Okay? It's difficult to see the most living cells through a light microscope because they are transparent, and we need to use a specific materials called cell stains, and these materials called chemical stains or dyes, to see the cells under the light microscope. Fluorescence microscopes or fluorescence, when dye gives off light of a particular color when viewed under specific wavelength of light. So we have a material called fluorescent materials. If we apply specific type of light, they will emit or give specific color under this wavelength. We have fluorescence microscopy makes it possible to see the location of the molecules and watch them move about in living cells. This is the light microscope. Now we have another type of microscopes called the electron microscope. Electron microscopes used to study objects smaller than 1 millionth of meter and use beams of electrons. So in the light microscopes we use a beam of light, while here in the electron microscopes we will use beams of electrons, not light, that are focused by a magnetic field. So these microscopes use a magnetic field to generate electrons. We have two types of electron microscopes. The first one, transmission electron microscope. The second one, scanning electron microscope. Let's study the transmission electron microscope. The transmission electron microscopes used to explore inside the cell. So we need to find the cell structures and large protein molecules. They produce flat two-dimensional image. So flat 2D image, while and this is the pictures of a result from the T electron microscope or transmission electron microscope. They are flat 2D, okay? While the scanning electron microscope used to scan the entire surface of the specimen without cutting them into thin slices, they produce 3D images of the specimen surface. So a surface of the specimen and the 3D image. So make sure to memorize the nature of the image for the electron microscope types, okay? And for the electron microscopes can be used to examine only non-living cells and tissues. You have to make sure that all of the specimens used in the electron microscope must be non-living cells or tissues, because samples need to be dehydrated and chemically preserved first before they place them under the electron microscope. So dehydration and chemical preservation will kill the cells, so the cells must be dead or non-living cells. Okay? And that's all about the microscopes and the history of the cells. Thank you and have a nice day.